How are you doing? This is a weird angle, but I thought, you know what? I've got these left over, so I thought I could just drag along the metaphor of the string to talk about polypeptide synthesis, you know, which is one of the cool things that DNA does in the first place. So let's do that. Let's say that this is our piece of DNA, right? It was double-stranded, but now it's just single-stranded because there's a gene right here that we want to use to make the protein that it encodes. So here we have it. We got RNA polymerase because the transcription produces messenger RNA. So we want RNA polymerase to connect here, say at the beginning, and start using the template to produce the mRNA that will have the complementary sequence to that of the DNA. And he reads it along, and as he reads it, I feel like this is a magic trick or something. As he reads it, it forms the mRNA that's just coming out now.
And he follows the gene. Ta-da! Woohoo!
So we formed an mRNA piece that is complementary to this DNA strand that can now go back to its home and whatever. And now we've got our mRNA that can go outside of the nucleus for eukaryotes, and let's see what happens next. Woohoo! Also, what happens in eukaryotes is that there are different bits on the mRNA that come together to form the complete gene, and then there are sequences in between the useful bits that don't actually form part of the proteins, of the subsequent proteins. So the bits that form part are exons, and the bits in the middle that have to be taken out that do not form part are introns. So say this one is an exon, and then from here to here it's an intron, and then from here to here it's another exon, and then from here to here it's another intron, and then from here to here is another exon. So you want all the exon bits to come together, and you want to cut out the introns. You want to just cut them out and just have the exons come together. And of course, when that happens, you get fragmented, and then you end up with a shorter piece of just the exons, exon number 1, next to exon number 2, next to exon number 3, and you have the final ready thing to be translated by ribosomes. And
that is such fun, isn't it? Right, let's get on to that. Let's do that.
So we've got the piece of mRNA. This is our ribosome. This is the ribosome. And we've got the piece of DNA that has just gone through the ribosome, right? Through the middle of it, and it's sticking out here. And it gets read inside the ribosome, and each codon, which is 3 bases in the mRNA that codes for an amino acid, is complementarily linked to a tRNA, and the tRNA has got the complementary bases. So if the mRNA has got a codon that is ACG, the tRNA will have on the other end the complementary bit to ACG, which is UGC. The tRNA doesn't just have the anticodon, but it has the actual amino acid that is associated with it, okay, on the other end. And that's what forms the polypeptide, right? And it reads it codon by codon, and say this has been read, this is the first codon, and we've got a little bit here. There we go, that's the start of our polypeptide there. Let's say it's like 3 amino acids. So 3 codons have been read, and it goes on, another codon, another amino acid added. This is the polypeptide that is now being formed, and this is the mRNA that is being read. So
read, and we've got the polypeptide coming along.
Okay, and this is done. And the mRNA can be recycled, and it can go off and do this again. And the polypeptide is released when it's complete. It does things. It probably needs to be folded. Of course, proteins have got, this is the primary structure, just the string of amino acids that's just been made. And then there's a secondary structure that it begins to form in, such as an alpha helix or a beta pleated sheet. And then we've got a tertiary structure, and then we've got a quaternary structure, and it's like bam! And then before you know it, you've got hair, and you've got like skin, and you're alive, and you're working, and you've got enzymes, and you're like, yeah, mother. And this happens all the time in everything. It's like the most universe. It's literally its life itself.
Because the tRNA has got U instead of T, I think. Oh fuck, I don't know.